Okay, welcome to MLG online classes. So in this lecture, I'm going to discuss about vaccines. So what are vaccines and why do we need vaccines and how vaccines will develop immunity towards any particular thing. Okay, can anyone say what are vaccines, students? Let me give you one example. So vaccines are the substances, antigens that will induce immunity. They will induce active immunity. Active immunity. So what is active immunity and passive immunity? So passive immunity, if you see the brief classification, we have two types of immunity, innate immunity and acquired immunity. In innate immunity, basic skin, mucus lining, all these are a part of inborn immune mechanism where they will protect towards all types of pathogens. Whereas acquired immunity is a specific immunity which is developed towards a specific disease or a specific agent. Say for example bacteria or viruses. So this is called acquired immunity, and acquired immunity can be gained by two mechanisms, either by active acquired immunity or passive acquired immunity. So what is active and passive, students? So I'm sorry, natural acquired immunity and I'm really sorry, natural active immunity and artificial active immunity. So what happens exactly in our body? So this is the body, then this is blood vessel, and say for example coronavirus. So coronavirus has entered into the blood system. So immediately after getting any virus, our body will start develops antibodies against it. So it will take some time to develop antibodies, and these kind of once development of immunoglobulins, IgGs, our body will become immunized to corona, right? So like this. So how to induce antibodies? Like this, okay? So in order to have immune resistance toward coronavirus, I need to give that same antigen to my body. I need to give the same coronavirus into my body. Then only I will get immunity towards a particular agent, right? So vaccines are the substances which are viral proteins or the same antigens which are given to the body to develop active immunity, to develop cell-mediated immunity, either cell-mediated immunity or B cell. B cells results in antibody production, whereas cell-mediated immunity means it will invoke both T and B memory cells to memorize the pathogenic pattern. So the substances which will evoke immunity is called as vaccines, and these vaccines are of four types. Before that, let me tell some interesting thing about coronavirus. Nowadays, corona became a very serious thing, a global pandemic, unfortunately. So what exactly corona happens means, whenever it is entered into our body, it will go to lungs. It will go to lungs and it will sit within the alveoli. In order to enter into alveoli, this coronavirus need a receptor. It need a receptor on the cell of the lung, and this receptor is ACE receptor, ACE2, angiotensin converting enzyme 2 receptor. With the help of this ACE2 receptor, our coronavirus will get entered into the body. So this is coronavirus. Coronavirus has three proteins. One is membrane protein, M protein, which is the membrane of the coronavirus. Then it has a crown-like structures. So what is crown? So these spikes are called crowns. These are spikes, S proteins. So S proteins are the crowns. Crown means like a crown. The king will have, right? Kings and queens will have this helmet-like thing. So that is crown. Since coronavirus has many crowns, it is called as crowned virus. Corona means crown virus. So corona has crowns. Those crowns are called as S spikes, and it has one more spike which is envelope protein. So it has E protein, M protein and S proteins. So recently what we understood is these S proteins are very important for accessing the cell. So it will use its S protein, particularly that is GRP, glucose receptor protein, some number 32 I think, GRP32. It will use GRP32 to enter into the cell. Okay, so how to develop vaccine towards coronavirus is, I will take only S protein. I will take only surface protein, because this S protein is involved in getting immunity, right? So I will take only S protein and I will introduce this S protein in the form of vaccine to the people. So when I induce this S protein to the people, definitely what their immune system will do, their immune system will start developing antibodies, and they will get antibodies against the coronavirus S spike, and they will become immunized to the coronavirus. Even if real coronavirus enters into their body also, this coronavirus will use S proteins. Already these patients has S antibodies, S protein antibodies, and they can immunized. So that is the concept behind vaccination towards corona. So definitely vaccine will take almost 1 year. In the fastest, as earlier as it will take around 1 year to develop. So it is really a concerning thing. Until then we need to be hope. Should not lose our hopes. Anyway, so these vaccines are of 4 types, students. So I can give 4 forms of vaccines. One is killed antigens, killed or inactivated antigens, then live attenuated antigens or vaccines you can say. Then we have subunit vaccines, then we have something called toxoids. So I have 4 types of vaccines available currently. Okay, killed vaccines, live attenuated vaccines, subunit vaccines and toxoids. So what are killed vaccines? Let me give you one example. The best example is our polio virus. So polio virus can be given in 2 forms. Either I can completely kill the virus and I can give a killed dead body into my body. So when I give killed dead body polio virus, body will think, okay, some virus has been entered into my body. So body will start developing antibodies towards the virus. So these kind of vaccines are called as killed vaccines. Whereas in live vaccines, we won't kill the virus or bacteria, rather than we will inactivate it. So it can't infect us, but it will still in a living condition. So these living viruses or bacteria will induce immune response and the immunity will develop towards these vaccines. Okay, so these are live attenuated vaccines. Attenuation means suppressed bacteria or suppressed virus will be used. Say for example, one good example is Mycobacterium tuberculosis. So there is a particular strain of tuberculosis bovis, and this tuberculosis bovis is a tuberculosis infection to the cows. So what we did is, this tubercle bacilli don't like potato starch medium. So whenever we are growing this bacteria in a potato medium, it don't like it. Okay, so it will suppress. So like that, in a unfavored conditions, we started growing the Mycobacterium bacilli for almost 13 years. So after, in the presence of bile, okay, in the presence of bile in that medium, we started growing this bacteria, and this bacteria completely lost its energy towards fighting against the immune system. It is still alive, but it cannot induce any pathogenesis. It cannot kill our body. So these kind of antigens can be safely administered into our body in the live form, and those still replicate in our body, but they won't cause any pathogenesis, because meanwhile the body will develop immunity against the pathogens. So these kind of vaccines are called live attenuated vaccines. Then we have subunit vaccines. Subunit vaccines means, rather than taking a dead protein or such as a dead polio virus or a live virus, these two are really, there are some conditions where we cannot even trust a dead virus. Say for example, corona. Can I give a dead coronavirus into the body? Who knows? It may reactivate, right? Because at last they are viruses and they are very clever. It became a pandemic. So I cannot go for, at any cost, I cannot risk to administer a killed coronavirus into body. So obviously I can't even induce any live coronavirus. So what kind of approach I can go for developing antibody in the patient? Maybe I can go for like this. This corona has 3 proteins, right? S spike proteins, coronal proteins, then it has a membrane protein and envelope protein. Maybe I will take only the spike protein. I will take the gene that is coding spike protein and I will insert this gene in a fungus, and that fungus will start synthesizing the spike proteins. So it is not the virus, but it is just a part of virus protein, a small protein, and I will give this protein as a subunit, and these kind of proteins are called subunits, and these subunits of the original virus protein will be given to any patient, and that patient immune system will activate it and it will develop immunity. So these kind of vaccines are called subunit vaccines. And last and finally is toxides. So what are toxides? So toxides are the toxic substances released by pathogens. Say for example C. botulinum. So Clostridium botulism is a bacteria that will secrete a toxin called botulinum, and this botulinum very dangerous. It will cause paralysis to the body. So what I will do is I will take the toxin that is secreted by the bacteria and I will give this toxin in a mild form into the body. So when after giving a toxin, a mild form of toxin, body will start producing antibodies towards the toxin and it will neutralize the toxin, immune system, and we will become immunized to the toxins. So this is how toxides will work. Toxides are the toxins. So let us see examples for each. Okay. So So let me say first is killed antigens. So killed vaccines, best examples are oral polio vaccine, oral polio vaccine, then hepatitis A vaccine, then we have one more, rabies. So these are the three types of killed vaccines. Okay, so killed antigen vaccines. Next let us see live attenuated vaccines. So live attenuated vaccines, one is smallpox vaccine. Then we have measles, mumps, rubella, MMR vaccine. Then we have influenza vaccine. All these are inactivated. Influenza. Then we have BCG, Bacillus calmette BCG for tuberculosis. Then we have yellow fever vaccine. So these are the examples for live attenuated vaccines. Okay. So let us see subunit vaccines. So the examples for subunit vaccines are hepatitis B vaccine, then Haemophilus influenza type B vaccine, Haemophilus influenza type B vaccine. Then we have meningococcal vaccines. They will cause meningitis, inflammation to the meninges, pia mater, dura mater and aragonite matter of the skull. Then we have one more vaccine which is human papilloma virus vaccine. Then we have pertussis vaccine. All these are subunit vaccines. Okay. Then toxides, best example is tetanus toxide, tetanus and diphtheria. Diphtheria will be used as a toxides, will be used as a vaccines. Other than this, students, I already shared with you my notes. We have a Indian national immunized schedule, vaccination schedule in the national world. So what are the vaccines that is mandatory for a growing baby? So we have some vaccines like vaccine and the booster dose and the method of administration. I shared these notes with you, just to go through it. Okay, so just you need to memorize this table. Almost 80% of this schedule is generally accepted throughout the world. So only some minor vaccines are varies depending upon different location, but most of them are used all over the world. Okay, so that is all about vaccines. Just go through it. Okay, thank you, students.